So I would like to welcome Dr. Thomas House from the University of Manchester, who is one of the co-investigators on the Juniper consortium. Thomas is a member of SPI-M and has been working with ONS on the COVID-19 infection survey. And I think he's going to talk a bit today about using that data to try and understand a bit more about household transmission of COVID-19, and in particular the sort of methodology that they're using to try and understand more about transmission from this data stream. Thomas is a really great person to follow on Twitter. So he's often putting out really useful pointers about COVID modelling work, and his blog is also really interesting to follow, which he's had a recent post about why is zero so hard. So I'll put links to those in the chat as well in case people are interested. So thank you very much, Thomas. I'll pass over to you now.
Hello, so are those slides displaying okay?
Yeah, that's great, we can see them.
Good, so thanks very much for the introduction, Kira, and yeah, I mean, I do also do a lot of whinging on Twitter and arguing like anyone sensible, but there's the occasional bit of constructive comment as well. So these are my affiliations, and an very amusing bit of nominative determinism. I do work on household models quite a lot. I'm one of relatively few people who does so, and a lot of my other interests are in how we can use kind of computationally intensive methods to bridge the gap between mechanism and data, right? So a lot of the kind of excitement in machine learning is that this lets us do things with complex data, and what I'm interested in is a bit of how we can get some of those, or certainly before the pandemic, I was interested in this in a more academic way, and now it's become much more timely, is how we can use sort of modern computational and data resources to fit models where we can really interpret what's going on, because there's a mechanism, and I'll say a bit about what I mean by that. So just a quick bit of history. Household models are really, everyone, you know, we teach undergraduates about the SIR equations, right? Lots of people have discovered or rediscovered these during the pandemic, but actually alongside those whole population models, there's always been this strong strain of household modeling in infectious disease, and that goes, well, the reason for that, I think, is relatively clear, which is that when you think about it, which is that you live with people in the household, and you have close repeated contacts with them. So within household transmission of disease is a very common route of infection. And in fact, one of the odd things about this pandemic has been it's maybe not quite so common as you would think. So sort of what tends to come out of other people's analyses is what are called secondary attack rates or secondary probabilities of infection, which are actually not that dissimilar from flu, but the overall population level R0 for coronavirus is much higher than flu. And I think that's something we still don't really understand fully, why the within household rates haven't scaled quite like the overall R0. Okay, so but they still play a role, right? Still a significant proportion of the infections that we're seeing are within household infections. Okay. But one of the things we noted in a recent SAGE paper is, like I say, that the fact that isn't 100% means that there's still something to play for in pandemic control with stopping infections within households, and we might come to that in the discussion. Then if you look at the demographics, most of the population, actually almost everywhere, they just maybe change what they call the household. They live in relatively small, relatively stable households. So you do get, you know, so it's a relatively unusual time being a student when you pass through lots of households with lots of different people quite quickly, right? Mostly it's quite a stable structure. And so if you're going to send people door to door, which is what the kind of real or shoe leather epidemiologists do, and then give us kind of mathematical epidemiologist data, but the kind of the real work in the trenches, well, if you've knocked the door of the household, you probably want to collect all the data from there, right? And as I kind of alluded to earlier, we can design interventions at the household level. So this pandemic, we've automatically done that. We've told people to stay at home, right? Other countries have tried out of household isolation, where someone's ill, you offer them a hotel room, or maybe force them to stay in a hotel. The way that these cheap LFD tests that are being distributed, the way they work, it's the parents of primary school children who use them, because they've really been tested in older populations. So it's high schoolers use them directly, and then the parents of primary school and nursery children use LFD tests. So that's leveraging the household element to make a mass testing work. And all sorts of other things, you know, the people who advocate for ventilation say, well, you open the ventilation in the home, don't you? Right? You open the window at home. That's where people are. So that's why they're important. Then a quick bit of the mathematical history, or kind of scientific history. I think there are really, this personal view, but I think there are kind of three eras in the literature. So Reed and Frost's work on households, prompted by the 1918 pandemic, was, I think, the first stochastic epidemic model. And I think they were actually must have been one of the first people to do Monte Carlo experiments. So apparently they used a modified roulette table so Monte Carlo being a method for, you know, based on generation of pseudo-random numbers, right? They used a modified roulette table to simulate infection in Baltimore households. And their work wasn't published at the time, but it was it's since been kind of documented and applied to, their data has been kind of reanalyzed with modern developments by Fraser and others. Then Norman Bailey was a kind of mathematician, then later I think worked for WHO. His book reviews a lot of the theoretical developments at the time based on Markov chain theory. And the empirical observations were almost all symptom-based. So Hope Simpson's classic work on childhood diseases, measles and so on in households. Reed and Frost themselves, looking at Baltimore households, all you could do is ask people what symptoms they'd had. Okay, and then we move into a kind of more end of the 20th century. So Frank Ball derived a very general final size formula, going beyond, that didn't require the Markov assumption. There were other final size results, but I think it's really the Ball formula that gets the most use. Then there was kind of Cheryl Addy, Ira Longini, Betts Halloran, various other people who did statistical work with that formula and generalizations of that formula that they derived. For example, on the Tecumseh study. So the reason I separate this out is that the computers at the time were limited. So I think, you know, you're trying to solve these equations that I'm going to talk about. That's going to be the main topic of the talk on, you're trying to solve these equations on what were kind of, you know, 1980s hardware. There are all sorts of memory and computational problems with that. And also the data is based on viral culture, right? So that's where you actually try and grow the virus in some blood or something in a dish, you know. And then I would say the modern era, 21st century era, has meant we have modern computational methods. So, you know, Demarest and O'Neill had a paper where you could, you know, solve a really, really general, or fit a really, really general age structure, different household model by kind of brute forcing things in MCMC. And that works really well, actually. And I think it's always important to see that, you know, that the theory doesn't develop on its own. That's kind of enabled by the fact that we have modern molecular techniques. So now, you know, you can test yourself with a swab, and we can amplify sort of, you know, DNA and RNA and sequence things and look for antibodies. And it doesn't require culture. And that enables us to be much more ambitious with what we do as theorists, right? But I'll just make this other point that, having said MCMC is brilliant, right? No one's really doing it because we're in an emergency. So what people have tended to do is go back to last generation's models, right? So we have all that. So we go, and that's what I've done. I've gone back to the ball final size equation, because you know that's mature and is able to be deployed quickly in a way that a really big, complicated MCMC sampler just isn't. Okay, so the ONS study. So the part of the point of what I said about, you know, you send someone to knock a door, that there are two big community studies of coronavirus in the UK. One is REACT, and REACT is run on the basis of posting people tests which they administer themselves. The ONS design sends a field worker in personal protective equipment to each household. And because you send them to the household, you might as well recruit each person in the household. Now, you could argue this both ways. So some people say, well, REACT, they sample one person per household, and that means they have a simpler noise structure. Because cases tend to cluster in the household, that means that the ONS numbers are noisier because you get more agglomeration of cases. On the other hand, people do live in households, so it doesn't lead to a biased estimate of prevalence, doing a cluster sample, and it means that we can assess transmission in the household, which the REACT study can't. But they're for different reasons. REACT is much more finely spatially resolved, so really it's great to have two designs. You know, most countries haven't got one. The fact we have two is, you know, I think one of the things we've got very much right in our pandemic response in the UK. So here's an example of what the ONS design looks like. And this design was kind of a compromise between what the ideal data collection would be and what's practical and affordable. So a household recruited, in this case, we've got participant ID 1 to 4 going up the y-axis, and they get 4 visits spaced about a week apart, and then they get visits for the rest of the year at more like monthly intervals. And here's an example of the kind of transmission tree you might see, or that might generate data. So I've drawn this red transmission tree going through the household, infecting participant 4, who then infects 3, who then infects 2. And what we see is the red pluses. So the blue crosses are negative swabs, and we see the positive swabs. Now, the problem is with this design, we don't know what order the infections happen. So it could have been, actually, you do sometimes see someone becoming swab positive before their infector does, or that can't be ruled out. So the fact that someone came first doesn't even tell you that. And quite often you'll get the whole household converting at once. So we don't know what order these events happened in. So what MCMC would try to do is integrate over all of those networks. But as I said, that is too slow. If you're me anyway, maybe someone who is a better MCMC coder. So the problem is, what do we do about that? Well, we have to compromise. And so what I've done is split the data into 4 time periods. And just to highlight some, I'm going to show data that's all been publicly released anyway. The actual data of the most interesting bit of this has not been cleared for release. At the time I booked this, I thought there's no way it won't have been cleared yet by the 12th, but you'll have to wait a day or two and look on archive if you want the actual numbers. So this is more of a methods, it's more of a methods talk, but I will show some data, and it's I&I, so I think we're all right with mathematical methods being the main focus. But the idea is you pick, so this was actually done on the English bit of the data, because it's these time periods are supposed to line up with policies. So there's April till September, when we basically had low prevalence, the schools were shut and no new variant. Then September till mid-November, there was high and growing prevalence, the schools open, but the 117 variant was not playing a big role yet. Then we had a period right at the end of last year with high prevalence, the schools, I should have said, that should say schools mainly open. So the schools mainly open and the variant emerged. And January to February, you have high prevalence, the schools mainly closed, and the variant being absolutely dominant. Okay, so there's a bit of thinking has to be done about how to associate each household with a tranche. And for the method we're going to use, that's you associate a household with the tranche in which it first had a positive. Then if that household has any positives, you kind of want to pull all of them into the tranche where that household is associated with, right? So I won't go through that too much, but it's kind of the right thing to do for this analysis, right? This kind of compromise analysis, which is not, like I say, if you wanted to do the most statistically rigorous thing, there are other approaches, but the errors of doing that are kind of controlled, right? Okay, so a bit of maths. What is a household? So suppose you have n participants and we index them with indices I and j. The square brackets on n is integers 1 to m. I've picked up that very space-saving notation from the machine learners I hung around with. It's a very nice notation from computer science that saves you a lot of writing. So these individuals are then in m different households, and the 8th household is a set of indices HA. And the point is that, so this is just fiddly notation, but all I'm saying is that each household is a subset of the population, so that the households don't share, they don't share any members unless they're the same household. And every individual is in some household. That's all those three conditions are saying formally, so that the households are partition of the set, and the size of the 8th household is NA. And then within such a household, we're going to let xit be a feature vector, or so-called covariates associated with the ith individual, and yi is going to be their test result, right? So we have x's, which are things like ages and whether the thing that you had was the new variant. And then there's, y is 1 if you got the disease and 0 if you didn't. Okay. So as I teach MSc students, before jumping to model, we should do some exploratory data analysis. So here's what these histograms are. ZKL is the count of households of size L in which there are K participants testing positive, right? So I sort of show these graphs and then people say, how do you define them? And I define them and I go, why is it so fiddly? So I don't know what to do in between that apart from just show it. But here's an example of the kind of histogram you get. And the important thing about these histograms is I've not shown the zeros because that's the most common outcome. That's enormous. But you get these kind of U shapes if you look on the bottom row, right, in the larger household. And even for the non-U shape, for the L-shaped histograms, these histograms are very, very much more variable. Their variance is very, very, very much higher than an equivalent binomial because prevalence is tiny, right? And the second bar, so prevalence is at like 1%, right? And the second bar isn't 1% of the first bar, it's much bigger than that. And that's because of transmission. That's the smoking gun for transmission events within the household that lead to that clustering. Although when false positives was the thing, lots of people are like, oh, they're all contaminated in the household, and that's where you get that kind of over dispersal. So these are for the different times. And you see that over dispersal pattern at all times. Okay, so then you can also do these density plots. These went out in SAGE papers. You can form a point in the plane in a kind of square for each household. And again, I'm just writing the definition out because people get annoyed if you don't define it mathematically. But the basic idea is I put a translucent square down for every household that has some infections in adults and some infections in children. And I put it at the proportion of adult and proportion of child cases, right? So a household in the top right has some adult and some child cases, right? And in fact, everyone's a case. Yeah, and there's no one in the bottom left because I'm only picking households that have some cases. Otherwise, the bottom left would just be enormous, right? Because most households have no cases when you look. And just to say quickly that the point is that people look at cases. Sorry, I've been talking about cases. What I really mean is positives, community positivity. And that's because the sort of free data that everyone's used to looking at is data that people that's picked up because people get symptomatic and order a test, and the point is we don't know why they do that or whether it's constant numbers of people who are equivalently ill do that over time. So a random sample of the population like this is that's why it's kind of worth all the effort, that otherwise you don't know what you're missing, right, through the existing case finding. Okay, and what this shows is that the children aren't, basically this shows that the children aren't immune, right? And part of the reason I did this is that people go, well, if you do a model that shows that children are bringing it into the households, is that just the model? And actually, no, it's there in the raw data that there are households where there are no adult positives, no only child positives. And of course, we could have missed the adult positive. They could have got a bad swab or something, but it really, you have to sort of think that this is pretty consistent with children often being the index case in the household. Okay. Then there's a kind of analysis that I really like. Oh, hang on, I didn't. All right, okay, ignore the equations. That's the wrong equations, but I won't try and copy and paste the right ones in. I knew there'd be at least one typo. Right. Okay, so you can look up what a residual is, right? Basically, a residual is how far the counts in a cell deviate from what would be expected under the null of non-independence. And what these are, again, these went out on the ONS website, I think, not the SAGE papers. These counts, and what you see is the B117 variant is associated with OR plus M on these, and the wild type is OR plus M plus S. And what you see is over the 3 time periods, the OR plus N type comes to dominate, and both are clustered in the households. And the other positives, so there are 3 targets on the PCR machine, and the new variant fails to trigger one of those pretty reliably. That's the S target. So it tends to be OR plus N. OR are the other ones. And basically something that fails other things, it's probably because you had a low viral load. So you don't expect those low viral load cases to cluster in households, and indeed they don't, right? That's what the residual analysis shows. So residual analysis is slightly more mathematical than the completely exploratory analysis that we showed, but it's about falsifying a null rather than fitting a model that's what we think is going on. So that's what is the main thing I've done with collaborators. So now let's have a think about how to model the within household epidemic, and that starts with this thing called the Selke construction. So suppose each individual I has a stochastic variable Ti for its infectious period, and that's picked from a distribution, and that each individual has a random threshold chi-I, right, psi-I, which is an exponential mean 1, a random variable, right? And that an individual then becomes infectious when their threshold is exceeded by the total force of infection they've experienced. So to see why that's equivalent to a standard definition of an epidemic model, you think about the conditional probability of an X1 random variable exceeding a force of infection that's changing over time, over a small unit of time. Yeah, and you see that it's what you expect. It's what you would expect from the definition of the random process of infection in like normal random process theory, right? So so having defined the Selke construction, you can then take a household H of size n, it's going to now convect to Y and a feature H X. Okay, and now suppose we've got a map iota that goes from all the 0, 1s up to onto some sort of integers, right? Then we can form a vector in the space of length 2 to the n vectors that contains a probability of every different possible outcome in the household. Okay, so the first element of P will be probability of 0, 0, 0, 0, no one gets it. Then 0, 0, 0, 0, 1, 0, 0, 0, 0, 0, 1, 0, right down to 1, 1, 1, 1, 1, right? So P is that vector. And it turns out that vector can be given by the solution of a set of linear equations, so a matrix that depends on model parameters theta, times that vector P equals a vector of all 1s, yeah? And the elements of B are given by this equation here, which is what Frank Ball derived. Okay, so omega is kind of the this nu and omega are things like that. They're strings of ones and zeros, right? And the point is that we want the nus to be all the sub-epidemics of omega. And the way you get to this equation, I won't go through it in the time, but you can actually get to it just by kind of logical reasoning. So you say, well, the probability of having a 0 is the probability that everyone in the household avoids infection from outside, right? So then the probability of having a 1 is the probability that everyone else avoids infection from outside, and that one person gets infected from outside. Yeah. Then the probability of getting 2 is either each one of them gets it from outside independently, or one of them gets it and transmits to the other, and then there's no other transmission events. So you have to account for all the non-transmission events and transmission events. As you go through that, just with basic thinking through, you end up with this set of equations, right? But it's a bit of a fiddly argument for talk. But believe me that if you are happy with there being probabilities of things happening, this is what comes out. Okay. So let's look at the components of that equation there. So the first one is the probability that you avoid infection from outside, which I'm calling QI. Now, this is kind of my group's our team's contribution. So it's Tim Kinyan Dewey who was first author on the paper. We've got an archive on this approach, and it's something with other people I've been looking at. So this is the bit that's sort of newish, what we say is that QI is e to the minus big lambda, where big lambda is your cumulative external force of infection. And then we want a way for that to depend on things like your age or whatever. So we use this kind of link function, which is the same as doing a kind of log linear regression, because you know that your force of infection has to be positive, right? But what you want is your alphas, which are the effect sizes for being a certain age. So alpha naught is the intercept, that's the baseline if everyone was the same. But you want the other vector alphas to be, in this top equation, there's a vector alpha dot the feature vector, yeah. You want them to have support on the whole of the reals, right? That's called natural parameters in statistics. And so that's why we need to use exp as a transform, okay? So the baseline individual is someone whose feature vector is all zeros, and your baseline probability of avoiding infection from outside is this little q, which is x to the minus x of alpha. Now, this equation 1 here, physicists call this the Gumbel, statisticians call it the complementary log-log link function, right? But the point is it has a kind of interpretation. So it's a way of bringing in linear predictors to this kind of complex household transmission model. So we'll report 1 minus q, and whether we do that as a kind of baseline force of infection or linear predictor or q is kind of a matter of taste, right? So the next component of model variability is the infectiousness at the individual level. And we normally think about that as arising from a distribution of infectious periods, which is what we'll do here. But really, it's just a measure of variability, the fact that people are there's just some extra difference between people that we haven't captured with all of the covariates. Okay. And we want the Laplace transform of this, and the parameter var theta is the variance of a gamma. So I'm going to pick a gamma. I'm going to say that people pick from a gamma. So this var theta is larger for more individual variability. Okay. So why is it the Laplace transform? Well, consider the Selke construction and assume that you're an individual with a Selke threshold psi, and you've got a baseline rate of infection acting on you lambda, That's going to be multiplied by an infectious duration t. So this is just, we've got a pair of people. One of them's got a psi, the other one's got a force of infection, big lambda, which is equal to lambda t. So the probability that you don't get an infection, because the probability you don't get an infection because the force of infection is too low is the integral over all values that t could have taken, times the big F is for density function and little f is for, big F is for probability function. There's a cumulative distribution function, and little f is for probability density function. And so it just turns out, because we picked the Selke threshold from an exponential 1, which was justified by the other equation, it ends up looking like a Laplace transform, right? So you get a Laplace transform of the variability in infectious periods out naturally from this kind of consideration, which is very, I mean, you can actually get it out from a slightly more abstract. There's some really nice algebraic probability that you can also use to get these results out. Okay. So the third component, anyway, so that's one component. That's the second component, was the individual variability, like on top of the covariates. And the third component is the infection rate from individual J to individual I. So again, we've got a baseline rate of infection internally, little lambda. Sigma I is a relative susceptibility. So it's bigger if you're more susceptible to infection. And again, we need to use a link function, so we'll use exp. And exp beta k is the relative susceptibility associated with the k feature. Similarly, we've got a transmissibility j, so some individuals can be more infectious, some can be more susceptible to infection. And that tau of j is, again, we use an exponential link to get a linear, to attach that to a linear predictor, which is gamma dot the feature vector. And exp of gamma k is the relative transmissibility associated with the k feature. Now, as you can see from that equation 2, you can interpret log of lambda as the intercept, or we can just leave lambda as a baseline transmissibility. N to the eta is a way of modeling the effective household size that's usually attributed to Simon Koshme. As you can see from that equation I've written there, that is the same as thinking of log n as being a transmissibility covariate. So you could also think log n is a feature. Okay, then to fit the model, so I can put all of that together and write a likelihood of the whole observed data, but there are actually 3 million observations, and I can only work on an extremely slow, secure Windows 7 machine or something. I can't remember, Windows Vista, or I don't know, some Windows version that's not very easy to use. And so a significant amount of the work is thinking about manipulating the data, which I've learned requires something quite serious like pandas or tidyverse or SQL or something. And actually the science of doing that is really interesting. I guess it's not strictly mathematics, but how you think about data and the relationship with data and what links. And if you've got visits within a person, within a household, how are you going to aggregate those? It is actually almost a talk in and of itself. And so we carried out this approximate Bayesian inference using the Laplace approximation, multi-restart numerical optimization. Now, the problem is, as I've said, the numbers haven't been released. You can see some other, I could have shown like the last iteration of this. So you get an effect size like, you know, children are a fraction as susceptible, or the, you know, children over a certain age are a certain percentage more likely to bring into the household. But that exact number is currently viewed as sensitive. Awaiting clearance. So, you know, like I said, a couple of weeks ago, I said to Kira, oh yeah, I've written this paper. It won't take very long to get the numbers out. You know, I'll definitely talk about this because I want to be so interested. And then Hofstadter's law is it always takes longer than you think, even when you account for Hofstadter's law. So that applied here. So thank you to the ONS team. The ONS are brilliant to work with. Actually, they're incredibly conscientious and very dedicated public servants, in my opinion, despite having just, you know, complained about clearance. That is, you know, it's something that arises from conscientiousness, which is good. I can't speak highly enough of the ONS as an institution. Also thanks to the Manchester colleagues, Juniper Consortium, other funding from Royal Society and UKRI, Alan Turing Institute, people working for SAGE, probably lots of other people as well, and everyone here for their time. Thank you.
Thank you very much, Thomas. That was really interesting. I'll give you a little virtual clap. It's always very difficult in these situations, you can't clap. So does while we wait, just to remind everyone, if they want to use the Q&A button to ask questions or the chat. So Sean has a question actually. How much information is lost by only considering the final infectious status of the people rather than their test positive times?
Okay, so a bit, but the risk is that by including times, you could get things really badly wrong. So that's I think why I took that approach, that like there is in theory quite a bit of information in everything over time. But if you think about it, each individual has a latent process where they get infected, their viral load goes up, their viral load goes down. They infect people, some people don't swap, you know what I mean? You'd have to start including a lot of other parameters. So I think you could get lots more information out than what I did, but you'd have to get some really big MCMC mixing to adjust for all those unobserved things that matter in the time varying model. And part of the reason that the maths is so satisfying for the Ball equations is that they're extremely robust against model misspecification. So I see that in a way that's a pain because it reduces what's identifiable. You can't learn as much using so it's true, you can't learn as much using a Ball equation type approach as you can using a general time varying approach. Yeah, but if you get your time varying approach wrong and you try to fit a time varying model, you'll get things really badly wrong. Whereas the relative insensitivity of the final size to other things about the model is in some ways a feature. It means less is identifiable, but there's less ways you can go wrong. So I think Frank didn't mind too much this splitting things up into tranches as a kind of compromise. He was kind enough to kind of review the approach. And I think, you know, it's the kind of thing you do as a practitioner, which is, I mean, you know, he had a paper with Lawrence Shaw looking at what you should really do with time varying data, but that, you know, if you need an answer in a week, that was also something that's going to be hard to do. So I don't know if that answers the question, but
that doesn't, Sean. You can always write a bit more of a further question in the chat. If I can abuse my chair position role to ask a question, and it was coming back to, so you mentioned about that children are not immune and how you showed the plots of the children coming in. So does that mean that this ONS data can be used to try and understand a bit more about what the role of children and young people are in transmission? And do you have any insights into?
Yeah, so that's what the reason I was put on this kind of tight leash with this recently, because it turned out to be quite big numbers when they were publicly released, that children were playing this big role and bringing into the household. And then because that really took off, they were like, oh, well, let's make sure it's right. This and you know, what will happen is the numbers come down a little bit. It doesn't, so I've just seen as well, Tom King's asked in the questions, like, does it, can we do the serial interval? Not really in this model, but there is an estimate coming for relative transmissibility. I think it relates to Sean's question. It's like time is a bit harder to, it's hard to tease out, and it's irregular sampling, and the people who've got the best data on the different variants of sample people extremely frequently. So there's the Harvard group who are, it's the Harvard group are, they've got these basketball players, and there's various places around. There's the ATTACK study where they've done frequent sampling. The issue, of course, is they're not probability samples. So I kind of think we need a Bayesian approach where we use the ONS to debias richer priors somehow, or something like that, where you you've got some, like the NBA players are far too young and healthy, right? But if you allow yourself some sort of modelling assumption about how that's going to change in a typical member of the community, that might be the better use of it. I think on its own, I'd worry about the estimates because of the non-dense sampling. And so what do you have people who don't participate? It's quite rare, so that they're just removed. Just remove the row, which is not quite right, because you might say, well, they're a bigger household, but you could argue it the other way, that they're, but it's a small enough number that, generally speaking, either the whole household does it or no one does it. So I'm sure you could argue it either way, but it in practice just sort of take the rows as they come, rather than try and spend a long time doing work on something that didn't make a big difference. But the plan is to really, spend years drilling into these data and work out, hopefully there isn't going to be another pandemic, but we definitely should plan for one. And part of that is going to be spending years drilling through this and saying, right, can we get the tools so that the tools are ready for extracting the maximally useful information from this kind of study design in the future. So the index case comes out from the Ball equations. So it's integrated over. Basically what those equations do is they let you into, I showed all the different networks. The Ball, if you think you want to integrate over all the possible ones with some weighting, right? If you write down Bayes' theorem or Bayes' rule for those, so you'll have a large number of discrete structures that are the different routes that the infection could have taken. Now, what MCMC does is integrate over those in Monte Carlo. So you get the computer to explicitly represent each network and add and remove links and arrows and so on in a way that lets you do that integral. But the Ball result is you can do that integral analytically. So it integrates over them, and then you can recover from that what the relative chances are. But for a given household, its predictive power is poor. So people say, "Oh, does it mean for that household, it was that one?" It's like, no, I've made a population level inference about what the rates of external introduction are, but it hasn't explicitly represented most likely networks for each household. But that's why it's doable in 3 million rows, right? I'm not going to try and MCMC 3 million rows if someone wants an answer in a week. But maybe we can have software that would. I mean, this is the thing, if we start developing it now, I've got every faith that we can have software systems that will do gold standard inference next time, which hopefully there won't be again, but
Or not for a while, maybe.
Yeah.
Does anyone have any further questions that they'd like to ask? I do have one. I have one further question, and it's related to sort of the generation time. So that are you going to use this data to try and understand a bit more about the generation time for COVID-19? And
yeah, so that's I mean, there are people going to be looking at that. I think the problem is we need to understand a bit about the viral dynamics as well. I think it's the thing that it's very hard to isolate. So there's this model people are calling the Larimore model, which is basically that your log viral load goes up at a straight line and then down at a straight line. And if you want to fit a mechanistic model there, in log space, all the ODEs do is curve the top of those straight lines a little bit. So you fit like something a bit like an SIR model to the dynamics of the virus within an individual. And so people have tried either fitting the ODE model or fitting the two lines to this more densely sampled data. And I think it's very hard. I think it's hard to understand the generation time without understanding that a little bit, because you need to understand that people's rate of infecting people goes up and down. And there is this thing where like you get a long tail of PCR positivity. So you kind of need to factor in that people are very, very unlikely to have infected someone late in their infectious period, and all sorts of other things. So I think it's doable, but it like I say, it's another.
There's another question coming about.
Yeah, so we did some sensitivity on that. we did do some sensitivity on that, and it's like you'd expect that you get this risk that if you've not, it's like with any epidemiology. So a lot of the conceptual work I've done is how do I relate these transmission models to biostatistics of linear predictors and relative effect sizes and so on, right? And depending on what you think, that's either obvious or clever. So some people go, of course, it's obvious to how you include the covariates, but then no one else has done it. So in a way that sort of lets you use all that machinery of epi and biostats and so on. But it probably was easy, but it needed doing. It's the same as all of that. If your model is really not capturing something, then you don't think attendees can see can you see them in the chat?
So I think that question was just asked to us, Thomas.
Sorry. Okay. Sorry. So there's a question of does the inclusions change if the tranches change? And the answer is basically yes, not terribly. I mean, we did sensitivity analysis of almost everything, as you'd expect. But if in general, if your model is misspecified, then you're going to get problems in the same way that if a linear regression was misspecified, right? You're going to get problems for all the usual reasons of epi and biostats. But kind of the reason I was trying to set up this covariate-based thing and linear predictors and so on is we could start bringing in the things that epidemiology and biostats people know about to do with adjusting for different effects and causal pathways and so on into this kind of household world.
Does anyone else have any further questions? No one else has any more questions? I should say that this talk has been recorded because I know quite a few people are away on holiday this week. So if you have colleagues who are interested, then the recording will be posted on the Isaac Newton Institute website, hopefully sometime this week. So you can watch it back. And the preprint, Thomas, did you say for this is coming out later this week with all?
Yeah, hopefully, fingers crossed.
Well, if you follow Thomas or Juniper on Twitter, I'm sure you'll be the first to hear about the preprint. So thank you very much again, Thomas, for a really great talk. It was really interesting, and thank you everyone for coming. And hopefully we'll see you at our research meeting, which is going to be next month. So thank you very much.